Just looking at the last section of biology now, Johann, I'm going to turn to you because you're an expert in this field. What do we know about the role of DNA repair defects, for example, BRCA1, BRCA2, ATM, etc., and the development of prostate cancer?
So the first thing to say is that our DNA is being damaged continually, hundreds of thousands of assaults every day by lots of different carcinogens, smoking, alcohol, ultraviolet light. And the reason we don't get cancer is that we have DNA repair processes that are amazingly intricate and highly complex and very precise. And cancer essentially happens largely because our DNA repair processes have not coped with that insult in some way, and those DNA repair defects have led to an accumulation of DNA damage that generates cancer. And this is quite pertinent in prostate cancer since we now know for aggressive lethal disease, 10 to 15% of patients have inherited germline DNA repair defects, and these families have higher risk of many cancers. For the BRCA type, we know that it's breast, ovary, pancreas, leukemia, and other cancers. So what we know now for sure, incontrovertibly, is that a high proportion of lethal prostate cancers have DNA repair defects. The proportion is much, much lower for the more benign cancers, much lower chance of heritable defects in the more benign cancers, and this confuses us for a long time. If you have a family history, you have a much higher risk of having a germline defect, but what's interesting is that 7% of our patients at the Royal Marsden, in a cohort of 150, with no family history, still had germline defects. And what also fascinated me most is that many of the men that were diagnosed with lethal prostate cancer in their 70s with no family history, still had germline defects. So actually, our assumptions on age of diagnosis and family history were incorrect. I think the biology is fascinating because if you have a defect in these genes, you change the cell's balance. The cell usually has balanced repair. So you have a balance between high fidelity repair, low fidelity repair, and that balance is key. If you now tip the scale so you have more non-homologous end joining repair of double-strand DNA breaks, that generates rearrangements. Prostate cancer is a disease of rearrangements and gene copy number gains, not a disease of mutations. And there's clear evidence that actually what you have in prostate cancer is an imbalance with high NHEJ repair, low fidelity repair, and low high fidelity repair, not only in these cancers with actually these germline defects, and in fact we now know that a third of somatic prostate cancers have these DNA repair defects. And there's actually other evidence emerging that other genes that are lost in prostate cancer, CHD1. We have just published a paper in Annals of Oncology that CHD1 loss also impacts DNA repair. It often associates with SPOP mutations. These SPOP mutations hyperactivate AR and AKT signaling. So you can imagine if you have a DNA repair defect and you have hyperactive androgen receptor signaling, you're driving transcription. That transcription usually needs repair to actually repair the DNA damage caused by transcription, which is part of transcription. And if you've got an imbalance, that repair is done through non-homologous end joining, and you get rearrangements at those androgen receptor response elements, and that drives prostate cancer generation. But critically today, these data will impact our screening, focused, targeted screening on populations at high risk of prostate cancer with these gene aberrations in blood or in saliva. This is going to impact therapy with not only late-stage disease therapy, but also adjuvant, neoadjuvant, PARP inhibitors, carboplatin, maybe even radiation-type therapies with radium, and we'll have to get more data. At this ESMO meeting, we've seen some really interesting data confirming the results of our data published in the England Journal by Pritchard et al. From the group in Spain, showing even in Spain, 9–10% of prostate cancer patients with advanced disease have these repair defects. And we now have data from many groups, so I think there is no doubt this is real.